Hi, I'm Dr. Williford. I'm a physicist and a physician, and this is an explanation of the Luminescence Hypothesis of Olfaction. The sense of smell is called olfaction, and shockingly, we don't know how it works. During COVID, I was asked why COVID patients lose the sense of smell. I discovered that we don't know how the sense of smell does work, so we can't explain why the sense of smell sometimes doesn't work. The mechanism of the sense of smell is not understood. I now have an explanation, and it is the Luminescence Hypothesis of Olfaction. I will first explain the problem, and then explain how this solves the problems of what we don't know. The beginning of this video has a lot of numbers, with the number of olfactory receptors, how many types of olfactory receptors there are, and the number of odors that we can detect. Some of these are hundreds of thousands, some are tens of thousands, some are millions, and it all happens in the time to inhale once, just for the sense of an odor. The beginning of this video explains the problem, and you may want to pause the video when there are slides showing the parts of all of these numbers. It all comes together in the second half of the video when the luminescence hypothesis of olfaction is presented. This is a summary explanation of the full article, which was published in the scientific journal Sensors. Let's begin with the anatomy. We are able to sense odors with olfactory receptors, which are located in the olfactory epithelium. This is a layer of tissue on the roof of the nasal cavity of the nose. When these receptors bind to odor molecules, a signal is sent to the brain, and we interpret this as the sense of smell. Here is the problem. There are only 400 types of olfactory receptors, but we are able to smell between 400,000 to 1 million different odors, and these are molecules that are detected by the receptors. Since there are so many more odors that can be detected than we have receptors, there must be combinations of olfactory receptors that the brain interprets as an odor. There are too many odors and not enough receptors, so there must be combinations of receptors for each odor. I'm not the first to say this, but I am the first to do the math of the combinations. There is an equation that calculates the number of possible unique combinations that can be obtained from a set. Here, the set is 400 types of olfactory receptors. The number of possible unique combinations of 2 of the 400 is over 79,000. This is not enough for all of the odors, so the set of ORs must be greater than 2. If an odor stimulates 3 of the 400, there would be over 10 million unique combinations. Since there are between 400,000 and 1 million unique odorants, there has to be a combinations of 3 receptors to have enough combinations for all of the odors. This is just like music, where 3 notes make a chord. Here there are 3 olfactory receptors that correspond to an odor. There is a set of 3 receptors for each of the hundreds of thousands of odors. To explain the problem, let's consider the environment of the olfactory epithelium with a mixture of 3 odorants present. Each odorant, call them A, B, and C, would activate 3 olfactory receptors each, which we can represent by numbering the 400 different receptors. Say odor A activates receptors 1, 16, and 200, odor B activates 42, 68, and 310, and C activates receptors 29, 160, and 380. There are these 9 olfactory receptors being activated simultaneously in the olfactory epithelium and sending signals to the brain to be identified as the 3 odors A, B, and C. But since they are all being activated at the same time, there are other sets of 3 of these which I would call false sets. For example, receptors 1, 68, and 150 in this example are all activated together, but they do not correspond to any of the 3 odors there. They may code for an odor that is not there, or the set of 3 can be meaningless. Remember, there are over 10 million unique combinations of 3 out of 400 receptors, and at most 1 million odors. So the vast majority of sets of 3 are meaningless. So the question is, how can the brain understand that there are only the 3 odors A, B, and C present? This is much more complicated to explain when we consider that with 50 million total olfactory receptors in the olfactory epithelium, with 400 types of receptors, there are 125,000 receptors for each of the 400 types. That makes up to 50 million. If there is even a low occupancy rate of 10%, this is 12,500 of each of the 9 types of receptors present in the mixture, which calculates to over 100,000 receptors being activated in the briefest of time by those 3 odors. Using the same previous equation of unique combinations, there are 84 combinations of 3 with these 3 9 receptor types for the 3 odors of the 400 total types of receptors. 3 of these 84 sets are the 3 for the 3 odors that are present, but there are 81 other combinations of 3 that are false sets that would represent nothing or even could be an odor that is not there. Let's take a second and review this because there's a lot of numbers. Because there are so many more odors than there are types of olfactory receptors, there must be sets of combinations of receptors activated for the brain to identify each odor. There are at least 400,000 and up to 1 million odors and only 400 types of receptors. There are not enough unique combinations of 2 of 400 for how many odors there are, so there must be sets of 3 receptors for each type of odor. When there are mixtures present, there are multiple combinations of 3, most of which are not a set that represents an odor. There would be over 100,000 olfactory receptors being activated at the same time if there are these 3 odors present in a mixture. No one has been able to understand or try to explain how the brain can identify only the 3 odors which are there when there are so many olfactory receptors being simultaneously activated. In this example, the brain is receiving over 100,000 olfactory receptor signals from the 9 types of olfactory receptors present, and can accurately and consistently understand this overwhelming input of information as that there are 3 odors present. This slide has an example of one false set, a combination of 3 receptors being activated that does not correspond to an odor, which is present. This slide shows that 81 of the 84 combinations of 3 from the 9 types of olfactory receptors of the 3 odors are false sets. This is 96% false sets. It seems impossible how the brain can interpret over 100,000 receptor activations with 96% of the sets being meaningless, or actually the 3 odors that are present. I will do one more example, and then we'll begin to the explanation of the luminescence hypothesis of olfaction. We know the brain can distinguish 30 different odors simultaneously present. With a mixture of 30 odors and 3 olfactory receptor types for each odorant, there would be 90 olfactory receptors for the 30 sets of odorants. Calculating a 10% occupancy rate, the number of activating receptors is over 1 million. The problem is orders of magnitude more difficult than this because the 1,125,000 is the number of activating receptors the brain would analyze to determine that there are 30 types of odors present. The brain simultaneously must determine that other combinations of olfactory receptor activations are meaningless. Again, using the equation for the number of unique combinations, there are 117,480 unique combinations of 3 out of 90. 30 of these represent the odors that are present, and there is another over 117,000 sets of 3 that represent either nothing or odorants that are not there. How the brain knows how an odorant is not there when each of the sets of 3 is being activated in the same time span as those of the receptor sets that are there has never been previously considered or discussed. There are far more of these false sets than real sets. In the example with 30 odorants, there are 117,000 false sets compared to the actual real 30 sets. That's an astounding 99.9% that are false sets. And in this context, there's over 1 million receptors simultaneously being activated by the 30 types of odorants present. Since any odorant molecule may dissociate from one receptor and immediately activate others, the numbers may be even larger than this. The brain is being sent over 1 million receptor activations at the same time and can experience this as the presence of exactly the 30 types of odors that are present. Amazing. With the number of molecules present in these mixtures, there are potentially billions of molecules activating millions of nerves that the brain is interpreting as groups of 3 for chemical identity. How all of this occurs in split seconds is astronomically incomprehensible. The next problem to consider is the one-to-one correspondence of one odor molecule to one olfactory receptor activation, as is presented in all other current models of olfaction, but is not in the luminescence hypothesis, as will be explained. Let's consider the structural binding of the olfactory receptors to odor molecules. Here, different portions of each of the 3 individual molecules of the same odorant must be bound by 3 different olfactory receptors of a set. This must be true for each of the 400,000 to 1 million odorants, where different portions of the structure are bound to 3 of the 400 types of receptors. We know that some molecules do have binding to more than one receptor, but that all 400,000 do is pure speculation. We have calculated that with a low estimate of 400,000 odorants and 400 OR types, that's olfactory receptors, then each receptor binds on average 1,000 types of odor molecules. Since each odorant set is 3, now each receptor must bind an average of 3,000 types of odors with a 1-to-1 correspondence. The olfactory receptors must be nonspecific enough so that each one can bind to 3,000 types of molecules. However, they must be specific enough so that each of the at least 400,000 odors binds to a unique set of 3. The chances that this is true reach the highest level of improbability. A much bigger problem with the 1-to-1 correspondence is the timing. The problem is that with each molecule activating one receptor, then the timing of the activating of the individual olfactory receptors of the set of 3 is random. The next time any of the odorants are present in the olfactory epithelium, the timing of the activation of individual receptors of the set would be different. The timing of the binding of these 3 separate molecules of one odorant to the corresponding 3 olfactory receptors would be influenced by many factors, including respiratory rate and depth, since this is the process that brings the odor molecules into proximity with the olfactory receptors for binding. There are expected to be variations in airflow patterns and perhaps eddy currents would also be present in the olfactory epithelium. Changes in concentration would clearly create differences in the timing for the 3 bindings. With higher concentrations, we would be expected to create decreased time between the 3 receptor activations. Temperature and other environmental factors may also be contributors. For at least these reasons, the interval timing of receptor activations are random events, and there cannot be consistent time intervals between the bindings of the 3 individual olfactory odorant molecules and the corresponding receptors. Without consistent interval timing, there appears to be no conceivable way the brain could interpret this information as the sense of smell. The brain cannot recognize a pattern when there is no consistent pattern. In the example of 30 types of odorant molecules present in a mixture, remember there are over 1 million receptors that are simultaneously being activated. With a 1-to-1 correspondence, the timing of 3 molecules of one of the odors activating 3 types of olfactory receptors, when there is inconsistent timing of the activation, and this is in the context of over 1 million activations of 90 types of receptors, would need to be explained if any of the other models of olfaction are true. But this is an unsolvable problem with the 1-to-1 correspondence of one odorant molecule activating one olfactory receptor, and this is the presented mechanism of all other models of olfaction. What is new with the luminescence hypothesis of olfaction is there's a 1-to-3 correspondence, where a single molecule elicits the response of 3 different olfactory receptors in such a brief period of time that a different odorant with a different and unique set of 3 receptors that is simultaneously present in the olfactory epithelium can also be distinguished by the brain. This complex combinatorial coding with consistently reproducible time dependence is well explained with an analogy to music and hearing. Music has 12 notes in an octave, including the flats and sharps. A chord is defined by 3 or more unique notes sounded together. 3 unique notes, simultaneously experienced by the ear, are interpreted by the brain as a chord. If the 3 notes are separated in time, they are not perceived as a chord, and if repeated attempts have inconsistent timing between the 3 notes, it will just result in random, incomprehensible noise. This becomes even worse if more than one chord is attempted, like a mixture with more than one type of odor, each with 3 unique notes and all separated with inconsistent intervals of time. It is the combination of a unique set of 3 notes experienced simultaneously that creates music. In very much the same way, 3 olfactory receptors experiencing activation together in time is interpreted by the brain as an odor. So with all of that as an introduction to the problem, the mechanism of the luminescence hypothesis of olfaction that provides a 1-to-3 mechanism of correspondence with a single odorant molecule activating all 3 olfactory receptors of a set in near simultaneous time, such that the brain interprets this activity consistently with a corresponding sense of odor, will now be explained. The first event is the binding of an odorant molecule to an olfactory receptor, as shown on the left of this slide. This is followed by an event of quantum biology. In quantum physics, electrons exist to exhibit a dual nature known as wave-particle duality. An electron is a particle of matter, but its behavior is like a wave. Electrons do not alternate between being a particle or a wave. Instead, they possess the properties of both simultaneously. However, an understanding of the wave-particle duality of quantum physics is not needed to understand the luminescence hypothesis of olfaction. There is only one event that needs to be explained, and that is the quantum event of inelastic electron tunneling. Electrons in a molecule can change locations on the molecule by disappearing from one initial location and immediately being present at another. They call it tunneling because the electron has moved. When it appears at the new location, it has a lower energy, and because energy cannot be created or destroyed, the difference in the energy of the first and the second locations is put into vibrations of the chemical bonds of the molecule. The molecule is then in a high energy molecular state. There are two types of energy events, elastic and inelastic. When energy is transferred and there is no loss of energy, it is called an elastic event. Examples of this are a bouncing rubber ball, a stretched rubber band, or a compressed spring. In these events, energy is stored and then released. When there is a difference in energy between the first and second conditions, it's called an inelastic event. An example of this is a car crash. So putting this together, inelastic electron tunneling in the luminescence hypothesis of olfaction is where an electron of an olfactory receptor changes location in the molecule to a lower energy state, which leaves the olfactory receptor in a high energy state, and it's well accepted that this does happen with olfactory receptors. However, previous considerations were limited to what happens to the electron after the inelastic electron tunneling. What's new with the luminescence hypothesis of olfaction is what happens next with the olfactory receptor now that it is in a high energy state. Luminescence is the emission of light from an excited electronic state of a molecule species. With the luminescence hypothesis of olfaction, the high energy state of an olfactory receptor after inelastic electron tunneling decays and releases the extra energy in three light waves called photons. We know that inelastic electron tunneling can produce photons, and this is fully explained in the article, but a calculation was done with how much energy comes from the inelastic electron tunneling, and there's enough energy for 3 photons with the energy in the infrared spectrum. When a molecule decays from an excited electronic state, the energy of the photons which are released is determined by the molecular bonds of the molecule. This is where the energy is stored, in the vibrating molecular bonds of the molecule. Since the chemical bonds of different molecules are unique, the energy of the photons released from different molecules, here the olfactory receptors, are also unique. The next step in the luminescence hypothesis of olfaction is that olfactory receptors are excited by photons and send signals to the brain. There has been a lot of research on olfactory receptors. The isolation of genes that code for olfactory receptors, which showed they belonged to the class of G protein coupled receptors, led to the award of the 2004 Nobel Prize in Physiology or Medicine. This shows the importance of olfaction research in the scientific community. This is important for the luminescence hypothesis of olfaction because rhodopsin is the photoreceptor of rod cells in the retina and absorbs light to mediate the first step of vision by activating the G protein transducin. Amazingly, the olfactory receptor is designated as a Class A receptor, for which is known as the rhodopsin-like family, the same family as the light-sensitive photoreceptor of rod cells in the retina. So very much like light travels through the cornea and vitreous to the retina so that we can see, in the luminescence hypothesis of olfaction, light travels through the sustentacular cells, which is in the olfactory epithelium, and then excite olfactory receptors so we can smell. This is a new explanation that olfactory receptors are excited by photons. We will now discuss the timing, and this is what solves the problem of so many random receptor activations with other theories that no one can figure out how the brain can recognize sets of 3 to identify an odor. The luminescence hypothesis of olfaction has 3 olfactory receptors activated at essentially the same time, that the brain does see sets of 3 in consistent timing to recognize an odor. With the luminescence hypothesis of olfaction, a single odor molecule binds to one olfactory receptor, and then after inelastic electron tunneling, 3 photons are released that activate 3 olfactory receptors essentially simultaneously. The event of inelastic electron tunneling occurs on the order of nanoseconds, where there are 1 billion nanoseconds in 1 second. Then the release of photons from a molecule after inelastic electron tunneling occurs 1,000 times faster in picoseconds, where there are 1 trillion picoseconds in 1 second. Once the photons are released, they travel literally at the speed of light to activate adjacent olfactory receptors of the set for an odor. The distance between the olfactory receptors in the olfactory epithelium is so small and the photons are so fast that the set of 3 olfactory receptors for an odor are essentially experienced by the brain as simultaneously within picoseconds. This side explains the concept of overlap, which is the problem with all the other theories of olfaction and is solved with the luminescence hypothesis of olfaction. It represents the problem of timing that was discussed. Here, the top row represents the one-to-one correspondence, where each odor molecule binds to 1 olfactory receptor. Each odor is represented by a letter, and for each odor there are 3 olfactory receptors. For example, for the odor R1 in red, there are 3 R1 lines. The bracket for R1 shows the timing of the 3 R1 receptor activations. During this time, there will be other OR, which is olfactory receptors, which are activated in a set of another odor. This shows 3 of the blue B1 receptors activated during the interval for the R1 odor. There is also 1 of the green G1 receptors activated during the same R1 interval. So during the R1 interval, the brain receives the information for the 3 R1 receptors, also the 3 B1 receptors, and 1 of the G1 receptors in this example. It has not been explained how the brain interprets these 7 receptor activations as only the R1 odor. The top has 2 red, 2 blue, and 2 green odors, each with 3 receptors represented. The brackets for each of these is the time for the receptors of each odor to be activated by 3 separate odor molecules, and with so many molecules of other odors simultaneously present, there is inevitably overlapping of the time for each of the 3 receptors of any one odor, with olfactory receptor activations of many other odors. Each bracket of each odor has 3 receptors activated during that time period. There is no pattern here that the brain can interpret as identity of an odor. Because there are so many more odors than the 400 types of receptors that exist, that led to the certainty that there must be 3 types of receptors to code for a single odor. With so many molecules of odors, tens of hundreds or thousands of molecules of different types of odors in a mixture, all randomly activating receptors, the brain would be overwhelmed with overlapping sets that there is no way this can be identified as the odors present. Now, the bottom row represents the 3 olfactory receptors activations for each odor present with the timing of the luminescence hypothesis of olfaction. Here there is activation of the 3 olfactory receptors of an odor set is so close in time that there is no overlapping with the activations of olfactory receptors from any other odor. The timing of the set of 3 is so brief that with no overlap with other odors, the brain would detect the set of 3 for chemical identity of the odor. This is a logical explanation of how the brain is able to distinguish real odor sets from false sets, because there is an interval of time where only true sets are experienced. This window of time must be very brief, since there are over 1 million receptor activations in the condition of 30 mixtures. Considering the mixture of molecules present in the olfactory epithelium with a mixture of 30 odorants, it would seem to be impossible with a one-to-one correspondence that the individual bindings of 3 separate molecules of an odorant to 3 olfactory receptors could occur without overlap in receptor activation timing with the binding of other odorant molecules from the other 29 odorants present. If there is overlap, as would be expected with a one-to-one correspondence, false sets would result, and the brain could not sort out all of this overwhelming random timing of a multitude of olfactory receptor activations. However, the luminescence timing of picoseconds does fit well with this analysis. This is sufficiently fast so there would not be overlap in receptor activations in a set time interval, leading to set identification by the brain and perceived chemical identity for an odor. So to summarize, there are too many odors that we can detect for the number of olfactory receptors we have. This means there must be a set of 3 receptors for each odor. With the number of olfactory nerves sending signals to the brain, when there are mixtures, the brain can never sort out all of the randomness of receptor activations in a time period to identify an odor. The luminescence hypothesis of olfaction solves this problem with this sequence of events. First, an odor molecule binds to an olfactory receptor. Then, with the quantum event of inelastic electron tunneling, the receptor is left in a high energy state with vibration of the chemical bonds. Then the receptor releases this extra energy by releasing 3 photons in the briefest of time, measured in picoseconds. 3 photons essentially simultaneously activate the 3 olfactory receptors associated with an odor, and the brain receives this information without overlap of other receptor activations for odors present in the mixture. Just like the retina detects light for the sense of vision, the olfactory receptors detect light for the sense of smell. The next step is to validate or test this hypothesis, and I will need to collaborate with scientists who work with live tissue of olfactory epithelium and olfactory receptors.